Howdy folks, Mr. Ulrich here. Sorry I'm not in class. Had to get my COVID shot. We do what we have to do. These are extraordinary times. Anyway, I'm going to use this video here to go over the cell parts for division, which is what we've been doing in class this week. When I come back next week, we'll finish up the protein synthesis stuff and go from there. Anyway, if you are watching this at home and you haven't had a chance to watch the using Cami video that I put together and posted, please take a moment to look at that. We're going to be drawing a diagram, well, labeling a diagram, so you need to be able to draw like arrows and stuff, and doing it on Google Docs, not so fun. And I'm finding that Cami is pretty cool for doing that. So take a moment, if you haven't watched the video, I'll still be here. Go watch the video and come on back. If you're doing this in class, of course, you got the piece of paper in front of you. So there you go. All right, so when you click on that, you'll get something like this. Again, I would suggest using Cami to open the cell parts for division. If you want to use the Google Docs, that's fine. It's, again, going to be hard to do the labeling of the diagram. And again, if you want to just print it up, do it on paper, I understand. Okay, so when you open that up, you get something that looks like this. All right, and right here, list as many cell parts that you know. Think back, harken back to ancient times, back in, what, 7th grade? Last time you talked about parts of the cells. Or if you've done it in class, I know some of the classes are still doing cells or haven't quite gotten to it yet. Anyway, however long it's been, think about some parts of the cells and what they do. Certainly refer to the diagram. Where's my pointer? Where are you? Where are you? Down here. Certainly refer to the diagram down here to give you some ideas on what's going on. Open yourself up a text box, all right, and write down some answers. We'll see what we get. Whoever's covering the class is going to hit pause while we get some answers put in here. Cool. Hopefully you've taken a moment to maybe even get some people to suggest different parts that you have. The top parts that we get, of course, are the nucleus. And here, let's make this a better font, shall we? I don't like Times New Roman. It's boring. Let's go Roboto. Oh, I like that. All right, all right. And we'll make this a little bit bigger so that we can see it. There we go. Okay, so nucleus. What does the nucleus do? Well, many of my students will come to me from middle school saying the nucleus is the control center of the cell. Not so much control center of the cell. Really what it's doing, the nucleus, that's where DNA is stored. And DNA is the information, the instructions for protein construction. So saying the nucleus controls the cell is kind of like saying that like libraries run the country, you know what I mean? Because libraries have all the information. Anyway, DNA storage, good. All right. Other parts that students often come up with, oh, let's see, cell membrane might be one, right? That's going to control what goes in and out. Okay. Let me make this a little bit bigger here so we can fit all that in there. All right, cool. Excellent. All right. Any other parts we got here? Let's see, cytoplasm. Literally, cell stuff, right? Cyto, C-Y-T-O. Oh, don't move the whole thing. No, not the whole thing. Just highlight the part that I want. There you go. C-Y-T-O. That is the prefix for cell. Plasm, like plasma. This is stuff. Works. Right? This is where most metabolism takes place. Spell place right, shall we? Ah, oh, look at that. Place. There we go. Cool. All right, maybe you got some other parts in there. I hope you did. In one of my classes, we even got to the mitochondria, which middle school answer, powerhouse of the cell. What we need to know is that cellular respiration takes place there. And there's a bunch of other parts that we probably could, that you may well have come up with. Maybe you came up with vacuole, right, for storage of stuff, really. It's food, water, cell sap, all kinds of crazy crud, and those vacuoles. All right, hopefully maybe even got some more. I hope so. We'll see. What we need to do is start labeling these cells. I'm actually going to switch over to a Jamboard to do this because I think I can work a little better with the Jamboard when it comes to drawing stuff. But it's the same diagram that you have, right? Plus my pointer is a heck of a lot better. Anyway, let's see. Circle, no. Pen, yes. And we can even make it into a different color here. Let's do it in red. Beautiful. Lovely. All right. So, some parts. Let's start from the outside. We already talked about the function of the cell membrane, but we can certainly label the cell membrane. Right here. Right, moving our way towards the inside. Right, all of this material here and all of the material. Oh, come on, give me another. There. All of this stuff here, this is all cytoplasm. And you can see there's chunky bits floating around in here. All right, cytoplasm, I refer to cytoplasm as the paella of the cell. All right, it's like stew. It's got all the stuff, right? All the organelles are floating in it. And, well, if you think about it, it's got all of the precursors of all chemical reactions and all of the products of the chemical reactions, all the intermediates of the chemical reactions, and all of the waste products of the chemical reactions, all the enzymes of all the chemical reactions. I mean, there's a lot of, whoa, there's a lot of stuff in there. Anyway, cytoplasm. All right, and then some students note this looks like fried egg. I call it the fried egg diagram here. Right, and here in the middle we have the prominent yolk of the fried egg. It's not the yolk of the cell. Now this gets a little bit tough to label, but now that I'm using colors, this might make it a little bit easier. So because we need to be able to kind of tell the difference between when we're labeling this outer envelope, the membrane there, better known as the nuclear membrane. Right, sometimes it's called the nuclear envelope, right? And that's going to be the boundary of the nucleus. It's going to contain the genetic material, the DNA, right? Now what we need to do as well is label the nucleus itself, and I'll use like these little brackets like this to kind of make it so you can see that I'm labeling the whole structure, right? This whole yolk of the cell. Right, is the nucleus itself. I'll move this down a little bit so it fits. There we go. Perfect. Nice. Okay, now when we look inside of the nucleus, it looks a little bit like a delicious plate of spaghetti. Right? And of course, the nucleus is the storage of what? Of DNA. That spaghetti is the DNA. Now, here's the thing. When we think about DNA in a nucleus, what letter do we often think about when we start thinking about the shape of the DNA inside of a cell? I'm going beyond the double helix, and I don't know, maybe you know what I'm talking about. I'm thinking about, of course, the X shape of a chromosome. And here I don't see that. And that's because, well, these aren't chromosomes. Not yet, anyway. It's still DNA, right? This is loose, stringy DNA, right? It's kind of like chromosomes that have kind of fallen apart. They're not all wrapped up tight. And that's important because that means that this chromatin, right, which is the loose, stringy stuff on the inside, right? This is chromatin. Man, I gotta spell it right. All right, loose, stringy, and more importantly, readable DNA. All right, remember, in order for DNA to be read, all of the data, the information, is that sequence of bases which is on the inside, and so it has to be untwisted and unzipped in order for it to be read. All right, and chromatin is that loose, kind of stringy form of DNA that can be unzipped and unread. Unread and be read. Now inside here, there are these two little things here. They look like meatballs. There's a variable number of these in cells. There's not always the same number. All right, but these are, I call them the little old nucleus. All right, just because they are the nucleoli, or nucleolus. Nucleolus? No, nucleolus. We'll spell it right. Got all kinds of fun stuff going on there. Fat fingers. All right, here we go. Nucleolus. I call it a little old nucleus because you take the word nucleus and you stick a little hole in there, right? Take that out. Nucleus, put it back in. Boom, nucleolus, right? And the nucleolus is where RNA nucleotides are assembled. All right, so this is the site of RNA synthesis, right, is going to take place in there. All the different RNAs, excuse me, are the nucleotides for all of the different RNAs are assembled there. And ribosomes are essentially assembled there. I don't know. Anybody get ribosomes up here? I don't know if anybody got ribosomes, right? Site of protein synthesis. Hope so, since that's what we were kind of talking about. Okay, so there we've got the yolk, and we've got the white of the egg, right, the outside. What are these little top hats here? These little two weird-looking top hat things, right? These are actually called centrioles, right? And they're kind of weird. See, inside of the cytoplasm here, inside of the cell, there's a whole network of stringy proteins, all right, that are kind of shot through here that make up what's called the cytoskeleton. You can't see them because they're too thin right now, but the centrioles are part of organizing those strands and bits of cytoskeleton into structures called spindle fibers. We're going to see those in a second. So these guys organize cytoskeleton into spindle fibers. Right. And I'm actually horribly overgeneralizing there. Spindle fibers? Nice. Spindle fibers. All right. I'm overgeneralizing their function here, as we are with most of these things. And I'm focusing on what they're doing here in cell division. Okay, so we're going to see those. Those are the major players there. Of course, if we move on to the next diagram, this is a cell that's dividing. Right? This is kind of a classic fried egg non-dividing cell. When we looked at our cheek cells, you got a chance to see a cell that looked kind of just like this. And by the way, all of these structures are the structures that we can see with a light microscope, except for these guys. I have enlarged them for your viewing pleasure. Anyway, let's move on here. Let's get to the dividing cell. All right. Now, some things that we can identify right off the bat with our dividing cell. Right. We got the cell membrane. We have the cytoplasm. Right. These are things that we have already identified. So let's just get those out of the way here for us, shall we? Cell membrane. Let's put one right here. Well, yeah. Cytoplasm. Okay, any other things that we can identify right off the bat? All right, I'm hoping that you see these X structures, and you remember me talking about the X structure of a chromosome before, and you're going, oh, here are the chromosomes. And you're right, these are chromosomes. So let's label this guy's chromosomes. These are double chromosomes. We'll talk about the difference between double chromosomes, single chromosomes in a moment. Well, not in a moment, but later on. Chromosomes. I'll just say it's one of them, because we're only labeling one. All right. Something else that you might notice, here are our little top hats. They are no longer right next to one another like they are here. Here they're right next to one another. Right now they're kind of spread apart from one another. Right? And since we know that these centrioles here, their job again is to organize the cytoskeleton into spindle fibers. Guess what those lines are? Hey there, centrioles. What I was thinking spindle fibers, because that's what those lines are. Oh, and I got kind of backed myself into a arrow problem here, didn't I? All right, let's see. I can work this out. Let's see. Let's go. We'll do it up here. Okay. Now, you got to be careful. A lot of students will look at these and see them vaguely in this kind of centralized, almost circular shape and go and see the DNA in there, the chromosomes, be like, nucleus! No, it's actually not a nucleus. All right, these are those spindle fibers that I was talking about. All right, they're actually strands. That's not a enclosed envelope. What the spindle fibers do, right, just like the cytoskeleton, among other things, provides a kind of a track for bits of the cell to move around on. The spindle fibers are also there to guide the movement of, in this case, chromosomes. If you notice, each chromosome, right where the X meets, it's called the centromere, there is a spindle fiber that's running through the middle of it. Boom, right? And each one of these, boom, 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 right? They have a spindle fiber that's associated with it. All right, these spindle fibers are involved in, they provide a mechanism for moving chromosomes. All right, well, wait, that's kind of crazy. Can you see that? For moving chromosomes. No, here, we'll move the cell membrane down a little bit. Come on, come on. There we go. All right, because remember what we're trying to do here in dividing cells, we're trying to come up with two cells. So I need some DNA on this side, and I need some DNA on this side. And what's going to happen is each one of these chromosomes is going to split, and one leg is going to go to one side, and one leg is going to go to the other side. Right? We're going to get into the whole process of that later on. All right, but what's important for us right now, if we're going to talk about how cells divide and the movement of chromosomes and all this kind of fun stuff, we need to have the language. We need to know the parts so that we can talk and other people will understand what we're talking about. All right, now, next thing we want to do, let's see, did I miss anything? Chromosomes. Oh, we should identify chromosomes and be a little more clear on this. Sorry. This is tightly packed, condensed, unreadable DNA. There we go. All right. Cool. What we need to do now is let's compare the two down at the bottom of yours. I think here we go. Yours is going to look a little bit like this. I put the pictures up above on this here. I'm in Cami again. I put the pictures up above so we can kind of refer to them. This is a classic T-chart. We're going to compare what they have in common and what's different. So what are some parts that the dividing cell has or does not have that the non-dividing cell does not have or has, right? So what are some differences between the two, right? I'm hoping you can recognize right off the bat that the dividing cell has an intact nucleus, right, where, ooh, and I gotta change that font, don't I? Yes, I do. I like the Mr. Roboto. Let's do that. Make this a little bit bigger. There we go. Intact nucleus. And in the I always do this. I always think about putting the, get over here. Intact nucleus over here. And let's put the no nuclear membrane here. All right, now we gotta be careful. Back up. I'm new to this Cami thing. All right, we're gonna get there. All right, so nucleus present, no nucleus visible. All right, other differences that we have. All right, maybe you noticed that there are spindle fibers here instead. All right, so spindle fibers. All right. No spindles. And because of the spindle fibers, Well, not I said not because of the spindle fibers. I was going to say the spindle fibers being present are the result of the movement of these centrioles. So over here I got the centrioles are apart from one another, where in the non-dividing cell, the centrioles are together. They haven't had a chance to make those spindles in between them. All right, and also very important is the form of DNA. So in this one I have chromosomes, all right, where in the non-dividing cell there is chromatin. All right, regardless of whether it's dividing or non-dividing, it's still DNA. Right, they all have DNA. Right, the cell membrane is intact, right? That's still got to be doing its job, making the inside different from the outside, right? It's still there. It still have cytoplasm, right? It's still there. So the cells are intact. There's just a whole lot of movement going around on the inside. Okay, I hope that helps. And if you have any questions, drop me an email. I'm going to see everybody in class on Tuesday, of course. Well, not in class, but in classroom. Other than that, again, be nice to the sub, and I will see everybody later.